Hi everyone, in this video I'm going to be talking about phylogenetic trees and trying to give you tips and tricks on how to do well with them. In this first part I'm going to be talking about how to interpret phylogenetic trees, and in the second part I'm going to try to give you tips and tricks on how to draw phylogenetic trees. So to understand phylogenetic trees, you need to know that it's a representation of evolutionary relationships between organisms or groups of organisms. And one of the first things you need to do is identify if the phylogeny or the cladogram is showing you how the organisms are different versus how they are similar. So an example of this would be a phylogenetic tree that's showing you the number of differences in cytochrome C, or they're showing you the similarities between organisms and another particular gene. So the first thing you'll want to do is kind of look at the table heading or the information on the phylogenetic tree to kind of get in the right mind frame. Am I looking at differences between organisms, or am I looking at similarities between organisms? Number 2, be mindful of branching and nodes. There's a reason why an organism is placed on a separate branch compared to others. That is because it either gains or loses something that the other organisms had or did not have. And the whole point of the phylogenies is to try to show you those relationships and when these characteristics arose or when they were lost in organisms. The next tip that I have to offer is looking for patterns. Keep in mind that organisms that are closer placed on the branches are going to be more closely related. So you can have two completely different sets of data, and understanding where that common ancestor is for that set of data is going to help. Let me give you an example. So let's say we have a tree here with the common ancestor indicated here at the bottom. When trying to look for patterns in closely related organisms, it's important to keep in mind that this group right here, or this cluster, is very closely related to each other, whereas this group over here tend to be more related. And the fact that this entire group over here is placed closer to the common ancestor means that these organisms evolved first before these ones. The next thing you want to take in consideration is how accurate is the tree. There's a couple of things to look at or look for when you're considering accuracy. Number 1, you want to look at what data was used to generate this tree. Now, before modern technology, trees were normally placed or put together based on structural features, but we find that this is now not exactly the most accurate way to build the tree. You can use besides the structural features, you can use DNA and proteins to make phylogenetic trees, and these tend to be more reliable than structural characteristics. I've even seen some phylogenetic trees made from common carbohydrates found within the organism or made by the organism, and even some based on behavioral adaptations. For drawing phylogenetic trees, that is something that is exclusive to the FRQ portion, so we're going to talk about those rules or tips and tricks as well. So let's say on an FRQ, College Board is now presenting you with data and you have to create a phylogenetic tree. Typically, they're going to give you a template on which to draw your phylogenetic tree, so make sure that you draw your phylogenetic tree directly on the template that's provided. You don't need to redraw it in the lined area. You draw it directly on what's provided for you. Number 2, you want to take a look at your data and determine whether or not you are looking at differences or similarities between organisms. And the rule to remember is the more differences between organisms, the less likely they are related. Now, typically what I like to do, depending on what type of phylogenetic tree they're asking, I like to identify the extremes of my trees. In other words, I like to identify what my outgroup is or who the outgroup is and who are the most common related organisms. One of the things I like to do is ask for the outgroup what sets the outgroup separate from the other organisms. In other words, what do all the other organisms have that the outgroup doesn't have? Now, your outgroup is always going to be placed closest to the common ancestor. And the most related organisms are going to be farthest from the common ancestor. So let's take a look at an example of a phylogenetic tree that was presented on the 2018 AP Biology FRQ exam. So in this example, we're looking at a protein called the lysed protein that is found amongst mammals, more specifically amongst bear species. Polar bears are highly adapted for life in colder climates around the North Pole. Brown bears, black bears, and pandas are found in warmer environments. Researchers collected samples of mitochondrial DNA sequences from several populations of bears and constructed a phylogenetic tree to represent their evolutionary relatedness. Now, this is shown in Figure 1, but we're not going to focus on Figure 1 for this question. A researcher studying adaptation in bears sequenced the nuclear gene encoding a lysosomal trafficking protein, lysed, in polar bears, brown bears, black bears, and panda bears. There are 7 inferred amino acid substitutions that are found only in polar bears. Mutations that cause similar substitutions in the human lysed protein are associated with Chadek-Higashi syndrome, an autosomal recessive condition in which pigment is absent from the hair and eyes. The researcher used the inferred amino acid sequence to build the distance matrix shown in Table 1. So this is the Table 1 that it is referring to. And notice again, it says amino acid differences. So in this data set, we're looking at the number of differences between species. And so remember the relationship. The higher the number of differences, the less related the organisms are. So we're going to take this data and place the organisms on the template that was provided on the AP exam. So for Part B, construct a cladogram on the template to represent the model of evolutionary relatedness among the bear species based on the different lysed protein sequences. Circle the position on the cladogram that represents the outgroup. So the first thing I'm going to do in this example is identify where the common ancestor is. So the common ancestor will always be at the very bottom or the root of the tree. I've labeled it as CA for common ancestor. Now for me, I find that the easiest way to build these phylogenetic trees is to first identify who the outgroup is going to be and find who the most closely related organisms are. Now, since this tree is based on the number of differences, you always want to make sure you check to see what kind of information they're giving you. Because it would be a very different scenario if this was the number of things they have in common. But since this is the number of differences, what you want to look for is the lowest number of differences. Those 2 organisms are going to be the most closely related. You're also going to want to look for which organism has the least in common with all the other, in other words, the most number of differences. And so looking at this data, I am going down and looking at, here is the panda, here is compared to the other organisms. Here's the black bear compared to the other organisms, the brown bear compared to the other organisms. And looking at these numbers, see how this is 8, 7, 1. All of these numbers are above 30. That would make the panda my outgroup. And so when you start building your tree, you put the outgroup closest to the common ancestor. Next, you want to put your most closely related organisms at the opposite end away from the common ancestor. So that would be these 2 right here. And at this node right here, these could alternate. In other words, you could put the black bear here, brown bear here, or put the brown bear here and the black bear here, and it's not going to change your tree. So we've already identified the panda. We've put the black bear, the brown bear. The last one we have to place is the polar bear. And in the instructions, they ask us to circle the outgroup. So I'm going to circle the panda. Although you could just circle the name or the word panda, and that would be acceptable as well. I hope this video was helpful. Thanks for watching. Bye.